Hi everyone, welcome back to the Bioinformatics School channel. In today's video, I am going to show you how you can map RNA Seq Reads to a reference sequence using the tool STAR. This video was made using materials from the Galaxy training platform. This is the original tutorial and I will leave the link to this page in the description box. This tutorial covered RNA Seq data analysis, but I will cover the mapping parts and the parts that shows how to map RNA Seq Reads to a reference sequence. There's also a brief description of the data here, so I suggest you read it. In this tutorial, we are first going to install the tools we will use. We will install fastqc, multiqc, STAR, some tools, and IGV. We are going to use Anaconda to do this. After that, we are going to download the example data. We will use paired end reads. So we will download the paired end reads, and we are also going to download the reference sequence, both the fastq and the GTF formats. After that, we perform quality control and then we will map the reads to the reference sequence. After mapping the reads to the reference sequence, we will also get the mapping statistics. We will also inspect the BAM file. So that's basically what we are going to do. Now, if you want to follow this tutorial, you need to make sure your PC satisfies these requirements. In terms of hardware, you should have a computer that has more than 8 GB of RAM. And you should also have a computer that has at least 8 cores. Okay, so the minimum cores I recommend is 8. In terms of software, you should have a PC that runs Linux or macOS. Okay, because the tools we are going to use, some of them work on only Unix operating systems. So you will need to use Linux or macOS. You should also have Anaconda installed. We are going to use Anaconda to install the tools. I have tutorials that show how to install Anaconda in Linux and macOS systems. So just make sure you watch those videos to set up your. The links to those videos are in the description box. I also have tutorials that show how to install the tools individually. So just check the description box and then use the videos that will be of help to you. So that's about it. So without further ado, let's start the exercise. Okay, I'm on the terminal now. What I will do next is to install the tools I will use for this exercise. I will use Anaconda to do this. Again, instructions for installing Anaconda can be found in the description box. Before we install the tools, we need to add channels. So I'll add 2 conda channels here. So I'll start with the first one, which is conda forge. So I'll say conda config add channels. I'll add the first one, which is conda forge. Let's add the second one, which is bioconda. So we give the same command and then we add a channel name, which is bioconda. So let's run this. It's done. Now we are going to create an environment and install the tools in that environment. So I'll say conda create-n and I'll say mapping. So this is the environment name. After that, I will indicate the tools I want. So I will install fastqc, I will install multiqc, I will install STAR, I will install some tools, and then I will also install IGV. So these are the tools. So first I create my environment, which is called mapping, and then I install these tools in that environment. So let's run this command to install the tools. Okay, so let's type Y to confirm. So conda will install all the packages that we requested. Okay, the tools have been installed, so we can proceed. What I'm going to do next is to activate my environment. So I'll say conda activate mapping. I'll also create a directory called mapping. So I'll say make the mapping. This is going to be my working directory. So I'll cd into it. So I'll say cd mapping. Okay, now I am going to download the example data. So first of all, I'll create another directory called data. So I'll say make the data. So I will download the fastq files and I also download the reference sequence. So let's first go to the Galaxy tutorial page. This is the page, and again, the link to this page is in the description box. So let's scroll down to data upload, this section here. Let's click it, and then we will see some links here. We are going to use the subsets here. Okay, these ones are smaller. Okay, so if you don't have a high NPC, or let me just say if you are constrained, okay, you have constraints in terms of computing facilities, then you can use these files here. These are smaller, so you can run them. You can run the commands using your normal laptop. So that's what we are going to do. So let's proceed. So we have fastq files here. These are fed and reads. There are two samples here, but I'll use one sample. So that means I'll use this and then this. So that's what you're going to do. So you can download by highlighting and then clicking it. So if you do that, you'll be asked to save the file. Okay, you can do it this way, but I want to download using my terminal, so I'll just get a download link. So I'll start with the first one here, which is the forward sequence. So I'll move to the terminal now. So on the terminal, I'll use wget to download, but you can use any other tool, so it's fine. So I'll say wget and then I supply it with the link. I would also like to remove this from the name. Okay, so I want to maintain just this one in addition to the extension. So as I've given this wget command, I will also give an option to rename this file. So I'll say -o. The O is uppercase, and then I supply the new name, which is this one, and I'll say .fastq sanga. So let's run this command. So this will download the file for us. Okay, so we have the first file downloaded. You can do an ls to check it out. So this is the file. Let's get a second file. So let's copy the link. So this is the second fastq file. So we'll copy the link. And then we'll move to the terminal to download it. I'll use wget. So I'll say wget and then I supply it with a link. Again, I want to keep this one. So I'll copy it. Okay. I'll keep this one in addition to the extension. So I'll say -o and then I give the name. Fastq sanga. So the file will be downloaded. Okay, the second file has been downloaded. So let's do an ls and you'll find that also here. So what we have here, these are fastq files. So I'll put all of them in a directory. So I'll say make the data, and then I'll say /fastq, and then I'll move everything here. Okay, I'll move the fastq file. So I'll say mv gsm star, then I'll move them to data /fastq. Okay, that has been done. So we have the fastq files. Now let's get a reference sequence. So let's go back to the Galaxy page. Okay, we are back to the Galaxy page. So we are going to get the download links for the reference sequence. So let's scroll down to the mapping section. So let's check. I think it's somewhere here. Okay, so we are in the mapping section. So here the GTF file for the reference sequence has been given here, the download link. Okay, On the Galaxy page, okay, if you are running this tutorial on the Galaxy page, you only need the GTF. But then, since we are going to run the terminal, we need to get a fastq file as well, because we have to build the genome index. Okay, so we need a fastq file and we also need a GTF. So we will get a fastq file as well. So this link, we will use it, but let's get a fastq file first. So we will go to the UCSC database. We are here. So I'll leave the link to this page in the description box. So when you are here, just scroll down, search for dm6.fa.gz, this one here. So that's what we will use. So I'll download on my terminal. So I'll copy the link and then I'll move to the terminal to download. Okay, I am on the terminal now. So I'll use wget to download the fastq file. So I'll say wget and then I supply it with a link. So this is the file. So I'll download it now. Okay, the file has been downloaded. Let's check the file. Let's do an ls. So we will see it here. Okay, this is a gzip file. That means it's compressed. So we have to uncompress it. So I'll say unzip and then I give the file name. So this will uncompress it. So let's do an ls and we will see this one here, dm6.fa. So that is the fastq file. So we will move this to another directory. So let's create a directory. So let's say make the data/ref and then we move the dm6.fa into that directory. Okay, that's done. Now let's download the GTF file as well. So the GTF file we will download using the link that has been made available to us here, okay, on the Galaxy tutorial page. Ideally, you should download both the fastq and then the GTF from the same database, okay? So that's what you have to do. But for this tutorial, I am just demonstrating the mapping activity. So I am just using a couple of databases here. But if you are going to perform genome mapping on your data, your research data, I will advise strongly that you download both the fastq and then the GTF from the same database. So that's what I recommend. Anyway, so let's continue. Let's download the GTF file. So I'll use this one here. So I'll copy the link and then move to the terminal. Okay, so I'm on the terminal now. So I'll use wget and supply it with the link. I want to be consistent with my naming. So because the fastq format has this name, this base name dm6, I'll also give the same base name to the GTF file. So I'll say -o, that's uppercase, space, and then I'll say dm6.gtf. So I'll download it. So wget will download the file and then rename it for us so that we get this one here. So let's run this command. Okay, the GTF file has been downloaded. So if you do an ls, you'll find that here. We will move this into the data/ref directory. So I'll say mvdm6 and then I'll move it to the data/ref directory. Okay, that has been done nicely for us. Okay, so we can do an ls into the data/ref directory just to check the file. So we have them there. Okay, so we are good to go. So let's proceed. What we are going to do next is to perform quality control. We will use this to assess the quality of the sequence data. Okay, so the data here refers to the fastq files we downloaded. So we will first run fastqc, and after that we will aggregate the fastqc reports using multiqc. So let's take a look one more time into the fastq directory. So we say ls data/fastq. So we have the data here. So let's proceed. So we will say fastqc, and then I'll say data/fastq star. That means fastqc should perform the analysis on all the files in this directory. Okay, and we have 2 files. So let's run this command. Okay, so fastqc is working. Okay, fastqc has done its work. So let's proceed. Let's check the directory again. So ls data/fastq, and we will find 2 outputs, okay, for each fastq file. So these are the files. So the HTML file here can be viewed, okay, can be opened, and then we can view the reports using our browser. So we can open it with our browser and then look at the reports that has been stored in that file. So we are going to aggregate the reports. I mean, the fastqc reports. We are going to aggregate them using multiqc. So we will do that, and then all the reports will be aggregated and then be stored in a single HTML file. So we can view that HTML file. So it makes it easy. If you run multiqc on your fastqc reports and aggregates everything, it makes it easy to make comparisons, because you don't have to click on each of these files and open them. Okay, sometimes it becomes cumbersome, especially if you have lots of samples. It becomes cumbersome to open them individually, check the statistics, and then try to also compare. So if everything is in a single file or is in a single report, then it becomes easy. So that's why we are going to use multiqc to aggregate the report. So let's proceed. So I will say multiqc, and I'll say data/fastq / fastqc *. So let's run this command. Okay, so multiqc has done its work. So we will have 2 outputs. We will have this one here. This is the reports, and we also have the data. Okay, so this is an HTML file. We will open this one as well. So let's do an ls, and we will find the outputs in our current working directory. So that's it. I'm going to open the files, okay, the multiqc reports and then the fastqc reports here. I'm going to open them with my browser. So let's do that now. Okay, so this is the multiqc reports, and when it comes to data fastq, you have the fastqc report. So I am going to open all of them, this, and then this. So I'll open them now. So I'll start with the fastqc reports. This is the one for the forward, and then this is the one for the reverse. Let's begin with the forward. So we have the summary here. I'll focus on the per base sequence quality. So I'll click it, and then I'll get this one here. So if you look at this summary here, we see that the rates are of good quality, and that is why the scores are high. You may be expecting some of the box and whisker plots here. Yeah, like you may want to see them here, okay, but then they are all clustered here. This is because we are not using the entire data set. So the one that we are using is what has been shown here, okay, and the one that we have been using, the scores are quite high, okay, so that means that they have good quality. Let's also look at the reverse. We also see that the scores are high, but some of them are in this region, okay, but in general the scores are quite good. So that's about it. So if, let's say, you have quite a significant number of the reads having low scores, then it indicates that those reads or those bases are of low quality. And so what you can do is to perform quality trimming, okay, So you can trim and then remove the low quality ends of those fruits. But for this tutorial, we are not going to do any trimming. We are going to proceed with what we have because they seem quite good. Okay, they seem quite good. But you can also try and then do the quality trim, but I will leave them as they are. So that's about it. Now, with the fastqc reports, we use multiqc to aggregate the reports. And so you can also take a look at the multiqc report and then look at what has been given here. Okay, so you can check the statistics here. We have some analysis here, so you can check them and then look at the quality distribution as well. So here we see this particular mean quality scores, and then they are all in the green region, so it indicates that the rates are of good quality. That's what this basically means. But if you want to get more information, if you want to get further explanation for the multiqc report, then you can check the tutorial here and then just check the quality control section. Okay, that's somewhere here. Yeah, so you can check and then look at the report here, and there are some statements here that tries to explain the outputs. Okay, it explains the results, so you can check that and then learn more. But I will skip this particular part and then proceed with the rest of the analysis. So let's proceed. So what we are going to do next is to perform our mapping. So let's move back to the terminal and then start the exercise. What we are going to do next is to map the RNA Seq reads to the reference sequence. So the reads can be found in the fastq files. Okay, so let's do an ls into the data/fastq directory, and we will find the files there. We have the forward and then the reverse sequences. Let's do an ls into the reference directory, which is data/ref. So we have the reference sequence here. This is the fastq format. This is the GTF format. If you are going to use star to align RNA Seq reads to a reference sequence, or let me rephrase it. If you are going to map RNA Seq reads to a reference sequence using the star aligner tool, then you need to first create a genome index. So let's go and take a look at the star manual. Okay, it has instructions that we can use to generate genome indexes. So let's do that now. So this is the page, and I'll leave the link to this page in the description box. So when you open the manual, make sure to scroll down to this section, generating genome indexes. So we have the basic options here. So when you call star, you also need to specify these options. Okay, so we have number of threads, we have run mode, we also have a genome DAR. Okay, so this is where the output files will be stored. You also have to specify the fastq file or fastq files if you have more than one. And then you also need to specify the GTF file. And then you also have this option. This one, we are not going to use it. We will use the default settings. I'll add another option also. So basically, this is what we have to do. So let's go back to the terminal and then generate our genome index. Okay, so we are back to the terminal. So let's first create a directory called index. So I'll say make the data/index. We will use this to store the output files. Now we are going to execute our star command. Okay, so we will create a script, and in that script we will put the star command that will be used to generate the genome index. So I'll use nano to create a script, but you can also use any text editor. So I'll say nano generates index.sh, and then I'll add my shebang line. Then the next line, I'll place my star command. So I'll say star, and I'll say dash dash run thread n. That's number of threads. I'll say 8. I'll move to the next line. I'll indicate the run mode, which is this. So the run mode is Genome Reads. Let's move to the next line. So we specify genome dar. Okay, that will be data/index. We have already created this directory. Let's specify the fastq file. So that is genome fastq files. So that will be data/ref/dm6.fa. Let's specify the GTF. So that will be sjdb GTF file. And that is data/ref/dm6.jtf. Let's add another option, which is genome sc index, and we specify 12. So that's what we have. So we are going to run this command. So let's save the changes. I'll also try to make download links to the scripts in the description box. Okay, so you can download the scripts and then use them for your own activities. So let's save the changes and exit. Okay, that has been done. So if you do an ls now, you will see that the script has been created. We now have to add execution rights. So I'll say chmod +x, and I'll say generate index.sh. So let's run this. Okay, now we can generate the genome index. So I'll say /, and I'll say generate index.sh. So let's run this command. Okay, now the process has started. So let's wait for star to complete its work. Okay, star has completed its work. So the genome index has been generated. So we can take a look at the output file. So if you do an ls into data/index, we are going to find the output files there. Nice. Now we can map the RNA Seq reads to the reference sequence. Okay. So again, we are going to put everything into a script and then run that script or execute that script. So we are going to map the reads. So let me just ls again into data/fastq, and then let's ls into data/ref just to look at the files again. Okay, so now we are going to map the RNA Seq reads to the reference sequence. So I'll create a script. So I'll say nano, and I'll say map reads.sh. That's how it's going to be. Now, before we write the scripts, we need to create a directory. Okay, so this directory is where a star will store the output files. So I'll say make that mapped. Okay, so now I can write the scripts. Okay, I can write the script and then execute it. So I'll say nano map reads.sh. I'll add my shebang line, and then I'll start with my star command. So I'll say star, and then I'll say -- run thread n. That's number of threads, 8. I move to the next line. I say -- read files in. So we have 2 files, the forward and the reverse. So I'll say data/fastq/gsm_1 fastq sanga, and then the other one will be what? Data /fastq/gsm underscore 2.fastq sanga. We also need to specify the directory for the index. So we say -- genome da, and that will be data/index. We also need to specify the output formats. We will use the BAM format. So I'll say -- out some type. That will be BAM. We also need to sort by coordinates. So I'll say sorted by coordinates. And then we also need to give a prefix. So the output files, what name should be given to them? Okay, the base name. So we say out file name prefix. So that will be mapped / I'll use the same base name, which is this one here. So that's it. So now we have everything we need. So we are going to save the changes and then execute this command. Okay, so again, I'll have to add execution rights. So I'll say chmod +x map reads.sh. So now I can run it, but you can do an ls just to confirm that the script has been created. So we have that here. Now let's execute the script. I'll say /map reads.sh. Okay, so star is now doing the mapping. So let's wait for it to complete. Okay, genome mapping has been done. So let's take a look at the output files. Okay, so let's ls into the map directory. So we say ls mapped, and we will see the output files. This is the BAM file. It contains alignment records. We also have 3 log files here. This one, this, and then this. And we also have this file, which contains information about the reference sequence. Now with the log files, this one contains mapping statistics. This one is the main log file, and it contains a lot of detailed information. And this can be useful for troubleshooting and then debugging. This one is a progress log file. And this is where star writes the progress. Okay, it writes the progress of whatever mapping activity it's doing at that time. So in an interval of 1 minute, that is every 1 minute, star generates a progress report, and the report is stored or saved in this file. Okay, so with the file output, I'll focus on the BAM file and then the mapping statistics log file. I'll start with the mapping statistics log file. It's a text file, so I can use the cat command to display the content. So I can say cat mapped, and then I give the file name. So let's run this command. Okay, the mapping statistics has been displayed for us. Whenever you perform a mapping task, one of the things you have to look for is the number of reads that's mapped to the reference sequence. Let's take a look at the uniquely mapped reads. This is the number, and in terms of percentage, it is 82.20. Ideally, we should have majority of our reads mapping to the reference sequence, and that is what we've seen here. So things are looking good. We can also visualize this mapping statistics report here. Okay, so what we are going to do next is to run multiqc, and then use that to aggregate everything here, and then generate a plot file. So we can do that and then view the plots. So let's do that. So I will say multiqc, and then I will specify the log file, which is mapped, and then I give the name. So let's run this command. Okay, the multiqc activity has been done. So these are the outputs. Okay, so that's what we are going to view. So we are going to view this using our web browser. If you do an ls, you will find them there. So we have this and we have this. So that's what we are going to look at next. So let's open the HTML file with our browser. So this is the multiqc report for the star mapping activity. Okay, so let's open it with our browser. Okay, so it has been opened. So this is what we have. Okay, so what we have here, you can use the information here to kind of interpret. Okay, so each color here represents a report, and we have them here. So this color is for uniquely mapped reads, and we can see that majority of the reads mapped uniquely, okay, to the reference sequence. We also have other ones here. So one cool thing about multiqc is that it's able to aggregate reports. So it's useful, especially in situations where you have lots of samples. It can be cumbersome trying to open the report for each sample. But if you aggregate all of them into a single document, then it's easy to look at them and then also do your comparison. So multiqc is a cool tool for doing that. So that's about it. So let's move back to the terminal and then continue with the exercise. Okay, we are back to the terminal now. Let's ls into the mapped directory again. What we are going to do next is to inspect the BAM file. So we are going to use IGV to do this. So we will open IGV and then load this BAM file and then inspect the mapping results. But before we do that, we need to index the BAM file. So we will use some tools. So we say some tools index and we specify BAM file. So let's run this command. Okay, indexing has been done. So let's do an ls into the map directory again. And you will see a file that ends with .bai. So that's the outputs. Okay, so this means that the BAM file has been indexed. So now we can open IGV and then load the BAM file. So we see IGV and we execute it. Okay, IGV has been opened now. So what we need to do is to first load the reference genome, the reference sequence. So we go to genomes here, genomes, and then you click on load genome from file. And then you locate your reference sequence, which is here, data ref, and then I select the fastq file. Okay, so I have selected it. So once it's selected and everything is okay, you have the name appearing here. Now let's load a BAM file. So we go to file, and then we click on load from file. Then we can select our BAM file, which is here, mapped, and then we select the file here. So select it, and then it will be loaded. So now we have successfully loaded the BAM file. We also have the reference sequence here, so everything is okay. So let's inspect the alignment records. So with this reference sequence, if you want to inspect a region, you have to select it. Okay, so we have the regions here. So you need to select the one that you want and then inspect the reads there. So let's use CHR4. That's chromosome 4. So if you select that one, for example, you have to come to the top right side here and then zoom in. So you click on the plus and then you can zoom in. So when you zoom in, you will see the reads. Let's zoom in a bit. Yeah. Okay, so I was playing a couple of things here. The gray picks here represents coverage. Okay, that's the coverage information. The number of reads that's mapped to a position. And then we also have this and this. These are the reads. And we also have some lines connecting these reads. Okay, so the lines connecting these reads, they are junction events. Let's go to the Galaxy tutorial page and it explains this therefore. So let's do that. Okay, so when you go to the Galaxy page, we have some information here that we can use. I've already talked about the coverage information, but you can click on the solution here. These are the questions. When you click on the solution, you will have the information there. So the coverage plot here indicates the sum of map reads at each position. And we also have the junction events, which I talked about. So the junction events are the connecting lines between some of the aligned reads. So we have some like this one, like this one, like this one. These are the connecting lines. So they represent junction events or splice sites. Okay, so these are reads that are mapped across an intron. That's what this indicates. You can do other visualizations there. You can inspect and look at maybe specific regions. For example, if you were to use the full data, we can move to this region and also inspect that place. Okay, but we are using a subset, so we don't have to do that. You can also look at this one, the sashimi plots, okay, by clicking on the certain region of interest and then checking the plot. But this is something that I'll cover in another tutorial, so just take note of that. But you can try yourself as well and then see what comes up. So that's what I have for you in terms of the mapping of RNA sequence to a reference sequence using STAR. Okay, so that's about it. So after you are done with the mapping, you can also look at getting the read counts. And that is something that I will encourage you to also do. If you want to know how to get the read counts after mapping a reads, then you can watch the video here.